Hello everyone, how are you doing? I hope everyone is doing good. So, welcome to another video of the chapter Reproduction in Humans. So, this is the second chapter that we are on. So, we are almost going to finish this chapter in a couple of weeks. 2 weeks more, and then we will be done with the chapter. So in the previous video, we had discussed about ovulation, menstrual cycle, the different phases of the menstrual cycle. In this video, so we completed that topic. In this video, we are going to study the structure of the secondary oocyte in a bit detail because we need to understand fertilization in today's video. So that is why we are going to see the structure of the secondary oocyte, the different membranes of the oocyte, of the secondary oocyte, and how the sperm will enter, what is the cause, what are the chemical reactions, biochemical reaction and the physiological reaction that will take place for the sperm to enter into the egg, which will bring about the fertilization. Okay, so we saw one scenario, that is menstrual cycle after the release of the secondary oocyte. So when the egg matures in the Graafian follicle, so it has two options, right? So if the sperm is there, then fertilization will take place. The egg which is released from the Graafian follicle, it will fertilize with the sperm, yes, and fertilization will take place. Okay, if there is no sperm available, then this egg which is released by the Graafian follicle, it will degenerate. Yes, it will degenerate, and along with the endometrium lining, uh, this all will become the menstrual fluid and the menstrual cycle will set in. Okay, so this we had discussed in detail in the previous video. If you have still not checked out the video, please check that out, and after that only you will be able to understand the fertilization. So this scenario we have already covered in the previous video. If the sperm is not there, so the egg will degenerate along with the thickened endometrium lining. The another scenario is what will happen if the sperm is there. So how the fertilization will take place, that we are going to see in this video. So before that, we need to understand the structure of the secondary oocyte. What are the different layers, or what are the different membranes of the secondary oocyte? Okay, so this is the mature egg. This is the mature ovum, which is now ready to be fertilized. At this stage, it is unfertilized egg or ovum. So when the sperm will enter, so it will get fertilized. Okay, so let's begin with this. So you can see the diagram over here. We'll begin with the first, the introductory part, that is the egg of the female, human female is non-motile. So the male gamete, which is the sperm, and the female gamete, which is the egg, which is in front of you, the ovum, out of the two, this is motile. Okay, yes, this is moving, and this is non-motile, and it is without any shell. Yes, generally we have the understanding the egg will be having a shell or a protective hard covering, but in human female there is no shell, so it is non-cleodic. Okay, so this is the first characteristic of the secondary oocyte. Next, we move to the first outermost layer of the membrane of the egg, which is known as vitelline membrane. So you can see over here this layer, this lining. Okay, you can see two linings, and in between there is the space. So that you will call it as the pre-vitelline space. It's like, you know, outer membrane, inner membrane is there, and then you have the outer membrane. Okay, so this membrane is the vitelline membrane, and the space in between is the pre-vitelline space. Okay, basically this is the first cellular covering. It is the first cellular envelope. You will see, you will say that there are another group of cells which you can see, and there is this one more layer. So we will come to that. That is not actually the part of the egg cell. That's not actually part of the ovum. These are extra layers, and they help in fertilization. How we are going to discover that step by step. Yes, so this we are discussing, the membrane is a part of the oocyte. The membrane is part of the egg, of the ovum. So what is that membrane known as? Vitelline envelope. So it's a structure that is surrounding the surface, outer surface of the plasma membrane. So you have the plasma membrane and then you have the vitelline envelope over here. Okay, understanding, all of you are aware that the animal cell, any animal cell will begin with plasma membrane. There is no cell wall. So you have the plasma membrane, and outside the plasma membrane you have the this vitelline membrane. Okay, then another layer that you see over here is these cells, which are somewhat, some are attached, some are detached, some are, towards the periphery. This region or this covering is known as corona radiata. We will come to this, just wait. So corona radiata. Basically, this corona radiata, you can see an irregular lining of these, of the layer of corona radiata. Mainly, corona radiata will help the protection, help in protecting the egg, that is the ovum, and it will provide vital proteins to the cell. Vital means important proteins, important protein molecules for the development of the egg, for the cell, okay, which is developing inside. Yes, so vitelline membrane, outermost covering of the plasma membrane. Then outside that you have the corona radiata, okay, which is protecting the ovum, and more or less it provides the protein to the cell, also some important proteins. Then you will see a layer in between. Yes, you can see these layers, this region basically. There is no cell, but some space is there in between the vitelline and the corona radiata. So this is a non-cellular layer. Basically, it is made up of a glycoprotein structure. So it's a glycoprotein structure. Basically, this zona pellucida layer, it prevents polyspermy. What do we understand by polyspermy is that the egg is attacked by multiple sperm. Yes. So zona pellucida will make sure that only one egg is entered inside the, one sperm is entering inside the egg. Only one sperm is entering inside the egg. Rest all of them are then blocked. So as soon as the one sperm enters the egg, then it's like the gate is closed of the secondary oocyte. There will be some structural and physiological changes wherein the membrane structure will change, and then no sperm will be allowed to enter the egg. Okay, so zona pellucida is that layer which will give this function, preventing polyspermy. It protects the oocyte during development and also prevents implantation in the fallopian tube. Another function of zona pellucida is that it will protect the oocyte as and when it is developing. Once the egg sperm is also there, so protection, some sort of protection will also be provided by this membrane, in addition to the corona radiata, which we just saw, that is protection of the ovum and also giving some proteins to the cell. And another function is to prevent implantation in the fallopian tube. So you have the fallopian tube, you have the uterus, okay. So the implantation has to take place in the uterus, okay, not in the fallopian tube. So because if the implantation takes place in the fallopian tube, then it will result in an abnormal pregnancy, something known as ectopic pregnancy. So this is not natural, normal, abnormal, then this has to be surgically removed. Yes, so the implantation has to occur, that is once the fertilization happens, zygote forms, it has to occur in the uterus wall. Okay, so to prevent any attachment in the fallopian tube, who is helping? Zona pellucida is helping. Okay, it is preventing implantation in fallopian tube. Fine. So this is the layers of the secondary oocyte: vitelline membrane, zona pellucida, corona radiata. These are the three layers that you need to know. Three membranes or three regions of the secondary oocyte. So now we move to the fertilization. Having understood the structure of the secondary oocyte, so in simple terms, what is fertilization? The fusion of the male and the female gamete is what we call it as the fertilization. Fusion of the male and the female gamete. So basically what unites? The DNA unites, okay? Because the egg is there and the whole sperm does not enter, only the genetic material, that is the nucleus, will enter. So the female nucleus, female genetic material plus the male genetic material, the nucleus, will fuse and then a zygote will be formed. So female, the secondary oocyte is half the number of chromosome. Sperm is also half the number of chromosome. N plus N will give you, it will retain the diploid condition, that is 2N, which will result in the formation of a zygote. Then this zygote will further develop, multiply, divide, and the ball of cells will increase, and this will develop into the embryo, which will further develop into the fetus, the baby. Okay. So in simple terms, this is what we understand by fertilization. Yeah. Now, let's dig deep into the steps of fertilization. Okay. So what happens? So that was the general understanding or definition of the fertilization. How step by step fertilization will take place. Number one, the sperm will come and it will make an entry. It will try to break this layer, and it will try to have an entry towards it will make a way through this corona radiata. Okay. Second step. Remember when we discussed the structure of the sperm. Okay, so the head of the sperm, inside which there is the nucleus, it has an acrosomal cap, okay, acrosomal tip, which has a lot of enzymes, hydrolytic enzymes. So these acrosomal enzymes will be released, okay, released to do what? To clear this zona pellucida layer, because now it has to make way now and go inside the egg. So it will release this enzyme, and certain part of this zona pellucida will be digested to clear the way. Okay, then these enzymes will digest that particular portion of the zona pellucida, and then that region will be now free. The sperm can move inside. Then this will further penetrate the granulosa cells and then zona pellucida. Okay. So there will be granulosa cells also, which are the remnants of the Graafian follicle. It is not represented in the diagram over here, but in the previous video we discussed what are granulosa cells also. Okay, the remains, some of the cells will be there, the remains of the Graafian follicle. Okay, so it will penetrate corona radiata, zona pellucida, and granulosa cells. Okay, by releasing hydrolytic acrosomal enzymes. Okay, so two enzymes that you need to know is, one is hyaluronidase enzyme. This will digest the hyaluronic acid that binds the granulosa cells together. Okay, so this acid, which is released by the head of the sperm, it will break the cells, okay, which are held together, binded together. So breaking of that layer will be brought about this enzyme. Another enzyme is acrosin. This is a protease enzyme. So whatever, usually the membranes are made up of proteins. Okay, they have a lot of proteinic structures in them. Remember, lipid bilayer. So you have the lipid molecule bilayer, and in which there is a lot of protein component also. So this acrosin, a protease similar to trypsin. So it will break the membranes of the egg. So you need to remember these two enzymes. Okay. This is the initial step in fertilization. Then what happens? You can see in the diagram over here, the whole sperm will not enter, only the part will enter, and then once it is inside, the head is inside, it will release its DNA material. So now let's see. This is the sperm. So this is the tail, middle piece, and the head. Okay. The nucleus is here somewhere, and this is the acrosome, which will have the enzymes. This is the zona pellucida layer. This is the plasma membrane of the egg cell, and something known as cortical granules. We are coming to this. We are coming to this. Okay. So you can see over here some dot structures. Step number one, it crosses the corona radiata. Step number two, it is releasing some granular structures. Basically, these are what? The enzymes of the acrosome, like hyaluronidase and acrosin. So after release of the enzyme, now this membrane got digested. So there is a way now. Okay. So now there's a way for the sperm. It has now moved inside. Fine. So sperm makes the way through corona radiata. Step number 1. Step number 2. Acrosomal enzyme will digest a portion of the zona pellucida here. And then step number 3. You can see now it has gained entry inside the egg cell over here. Okay, the head is now penetrating inside. So sperm will bind to and fuse with the oocyte plasma membrane. So the membrane, outer membrane of the sperm and the plasma membrane of the egg, both will now bind. Okay. Yes. So these are the initial steps of the fertilization that we just discussed in the previous slide. So this is with the help of the diagram. Okay, I hope you can comprehend and things are much clear now. Step number 1, making way through the corona radiata. 2, releasing of the enzyme, which will clear some portion of the zona pellucida. Then both the membrane of the sperm and the plasma membrane of the oocyte will be binded to one another, fused, so that now whatever the inner DNA material is there, it will be released. What happens after this is, so the sperm has reached up till here, till the plasma membrane. Then in the next step, once the sperm binds to and fuses with the oocyte plasma membrane, sperm nucleus will enter the cytoplasm of the oocyte. Yes. So plasma membrane binding has been done. Now the inner nucleus will enter the cytoplasm of the oocyte. We saw in the previous diagram that there are some granules which is located near the plasma membrane. They will also release enzymes. Okay, they will also release enzyme. Okay, then the zona pellucida will become fertilization membrane. So now the sperm has entered, zona pellucida will undergo certain changes. Now it will not allow another sperm to enter, because now the zona pellucida becomes the fertilization membrane. Now both the nuclei are in the cytoplasm of the egg. So the sperm and the egg pronucleus are together in a nuclear envelope. Okay, like this. So let's just summarize. After the sperm plasma membrane has fused with the egg, sperm has been fused with the egg plasma membrane. So step number 1, okay, once fusion has been done, the genetic material, that is the DNA, has been released. So what will happen? Remember when we were doing oogenesis, so I told you only when the fertilization will take place, then the secondary oocyte, which was arrested, okay, which paused its cell division at meiosis 2, will now complete. Meiosis 1 will now complete. So after meiosis 2, what will happen? Meiosis 1, what will happen? Meiosis 2 will take place. So completion of the remaining cell division will take place. Okay, forming the ovum and the second polar body. Yes. Then this polar body will move towards the side. Okay. Now both the nucleus are ready to get fused. So just before the fusion, they swell. At this stage, they are known as pronucleus, not nucleus, but pronucleus, swollen ovum nucleus. And polar bodies will move towards the periphery. Okay? Yes? Sperm and ovum nuclei will swell, forming the pronuclei. Then in the third step, what will happen? They are coming closer to one another. The male pronucleus and the female pronucleus, they are coming to one another. Fusion will take place. Okay? Fusion will take place. So for fusion, the mitotic spindle and the centrioles, these protein structures will be required. Okay? Once they fuse, now they are known as zygote. So they intermix, the chromosome or the genetic material intermixes. Fertilization is completed now. DNA will replicate in preparation for the first cleavage division. So now after this, Once the zygote has been formed, now cell division will start, will take place, that we will see in the next video. Clear? Okay. So zygote has been now formed. Uh, So a bit detail regarding the entry of the sperm and how it blocks the membrane for other sperms to enter. Remember, we were talking about the cortical granules. So they release enzyme. What is their role? So that we will just see in a nutshell. So after the sperm is there, it attacks the egg. So like the kind of summary we are doing in this slide. So it attacks the egg. So this is the first step that we saw. Okay, the sperm is trying to make way through the corona radiator. These are the three membranes. Okay, this is just a summary that we are doing, a recap of fertilization. Okay, a recap of fertilization. Step number one, with the help of the acrosomal enzyme, the sperm cell will make way through the corona radiator and it will pass the granulosa cells also. Okay. There are some of the granulosa cells which are there in between of the corona radiata. Okay, granulosa cells of the corona radiata. Okay. Then step two. Yeah. Step two, what is happening? Acrosomal reaction, that is binding of the sperm with the zona pellucida. Okay. Binding of the sperm to the sperm binding receptors in the zona pellucida. This releases calcium ions in the sperm to rise. So when the sperm reaches the zona pellucida, okay, when there is a interaction between the zona pellucida and the sperm gets a signal to release more and more calcium ions. This will trigger, do why calcium ion levels has to increase? For triggering more acrosomal reaction. Okay, then this will digest the zona pellucida. Basically, a hole or something will be created in the zona pellucida so that the sperm can move forward for the fusion with the plasma membrane. So, clearing a path to the oocyte membrane. So till here the sperm has reached, now binding. So the sperm's membrane will now bind to the sperm binding receptors of the plasma membrane. Okay, all these are receptor guided mediated movement. So sperm crossing the corona radiata granulosa cells, then reacting with the membrane receptors present on the zona pellucida, then next moving inside, moving more further inside to the plasma membrane of the oocyte cells, and then you see these granules over here. We are coming to that. So step number one, corona radiata, making way through corona radiata. Step number two, making way through zona pellucida. Okay, here by releasing enzymes. Here also by releasing enzyme, and this will trigger calcium level increase, which will make the acrosomal reaction more faster and more powerful, so that now there is a hole or a portion, a way created for the sperm to move inside. Then step number three, the plasma membrane is now in contact, so the sperm will bind to the receptors on the plasma membrane, and then the fusion will take place of the plasma membrane. Step number four, fusion, actual fusion. The sperm and the oocyte plasma membrane fuse. Both of their plasma membrane will fuse. You can see this region over here. Okay, you can see the region over here. I will first clear. So here the entry has been done. This membrane of the sperm will break open, and now this portion will be fused, so that this purple color genetic material has now way to go inside the cytoplasm. As and when the fusion takes place, immediately what will happen? This membrane will be blocked. Okay, so that another sperm which is behind, so they will not have an entry inside the egg. So entry of the sperm contents, as soon as the sperm inner material is inside the cytoplasm of the egg, calcium level increases in the oocyte cytoplasm to rise. Now calcium level will increase where? In the cytoplasm of the egg over here. What will this calcium do? This will trigger something known as cortical reaction. Cortical reaction. What cortical reaction? These cortical granules are there. They will release some granules, some substances, which will cause this zona pellucida to become thick or to become hard. Okay, which will cause the zona pellucida to become thick or it will become hard. So as a result, the zona pellucida will harden and the sperm receptors are turned off or clipped off. This will not allow any other sperm which is gaining entry. You can see the sperm now moves away from the zona pellucida because no receptors and the zona pellucida has become hardened. There is a change in the membrane physiology. So this sperm will now go back. Okay, अभी entry इसको नहीं है like that. So that is where the cortical granules will come into picture. They will start releasing certain granules and this will make the zona pellucida membrane very hard and which will prevent the entry of multiple sperms inside the egg. Okay, when will this happen? As soon as the genetic material of the sperm enters into the cytoplasm of the egg. Okay, then they will release calcium ion and as a result of the increase in calcium ions, the cortical granules will start releasing the, they will start releasing cortical substances and this will prevent multiple entry of the sperm, that is polyspermy, because of the hardening of the zona pellucida. That's all regarding the fertilization. So in this video, we covered the structure of the secondary oocyte. Yes. Now we are clear which membrane has which role in fertilization, and some are there for preventing polyspermy. Zona pellucida, corona radiata is more giving protection to the cell. Vitelline membrane is also giving protection to the secondary oocyte. Then we saw general understanding of fertilization and the secondary oocyte when oogenesis, when we were discussing. So it is arrested at meiosis 1. So once the fertilization takes place, the remaining meiosis 2 will happen. Okay, these are the enzymes that will participate mostly in breaking the membranes for the sperm to move inside the egg cytoplasm. So step 1, 2, 3 we discussed. Then fusion of the plasma membrane of the sperm and the oocyte. Okay, then how the nucleus turns in, basically how the genetic material swells and becomes pronucleus, and then they come together, form mitotic, spindle and centriole protein structures for their fusion, that is genetic recombination. Zygote will take place, zygote will be formed. Okay, and this is detailed steps of fertilization with the help of diagram. Okay, so in this summary, we just saw two detailing, that is the increase of calcium ions and hardening of the zona pellucida by the release of the cortical granules. Fine. This is the assignment regarding this topic. You're going to write it and submit it on the drive. So that's all in this video. In the next video, we are going to study the next step, that is embryonic development. Because now we know the zygote has been formed. So it's like a story. Zygote has been formed. Now what happens to the zygote? So it undergoes embryonic development. Okay, so how the embryonic development takes place, what are the stages, that we are going to see in the next video. So the first stage we will discuss cleavage in the next video. The first and the second, sorry, cleavage and blastulation. Till then, keep revising. If you have any doubt, make a note in your notebook and you can ask me in the live class. We will discuss that in the live class. Till then, take care. Thank you. Bye bye. See you in the next video.